Hey guys, welcome back. This is Lei. In this video, I'm going to show you how to analyze CRISPR screen data. Before we dive into the data analysis, let's first understand the technology that makes it possible. At the core of CRISPR screen is the CRISPR-Cas9 technology, a Nobel Prize-winning technology that allows researchers to precisely edit a gene in a living organism. This system uses the guide RNA to direct the Cas9 protein to a specific DNA sequence, where it makes precise cut, effectively disabling the target gene. A CRISPR screen harnesses its power on a massive scale. Instead of editing just one gene, we create a diverse pool of millions of cells, where each cell has had a single different gene knocked out. After applying a selective pressure, like a drug, we're left with a powerful dataset. The challenge and the focus of our talk today is now how to analyze the genetic information from the surviving cells to pinpoint exactly which genes are critical for the function we're studying. There are two experiment designs for CRISPR screen: the pooled CRISPR screen and the arrayed CRISPR screen. In the pooled CRISPR screen, you take a million of cells, each with a different gene knocked out, and grow them all together in one big pot. After applying a stressor, like a drug, you use sequencing to see which gene knockouts survived or died out, revealing the key genes. An arrayed CRISPR screen is a more organized approach, like a library. Each well on the plate contains a population of cells with the exact same gene knockout. This allows for more detailed individual analysis, often with a microscope, but it's much more labor-intensive and expensive. In the real world, the vast majority of high-throughput genetic screening data you encounter comes from the pooled CRISPR screen. Depends on the goal of your study, the experiment design can be treatment versus control, early versus late, or different time points. You will need biological replicates for reliable statistical analysis. You can, but you don't have to have technical replicates. The selection types can include negative selection, which genes are depleted. These genes are usually essential genes. Positive selections, which genes are enriched. These can be protective genes. Resistant screens for drug responsive analysis. The data type you get from a CRISPR screen includes FASTQ files, single guide RNA library file, and a sample information table. The workflow of the CRISPR screen analysis is very similar to the one for RNA-seq analysis. The steps include read counting, statistical analysis, basically it's a DE analysis, quality control, functional analysis, the goal enrichment analysis, and visualization. The most important tool is called MAGeCK. It's a command line tool. You can perform quantification and statistical analysis with the same tool. The results are stored in spreadsheets that can be opened in Microsoft Excel. You can use R to perform QC, filtering, and plotting, but it's optional. Let's start by setting up your analysis environment. You need a Linux system, but it doesn't have to be powerful, because our CRISPR screen datasets are small. Even your laptop can handle it. Now open your terminal. Create a conda environment for MAGeCK. Activate it. Configure the conda channels in this order. Install MAGeCK. You don't need SIGTAG, but this package is very powerful for manipulating FASTQ files. On the other hand, open your RStudio. Install all the essential R packages. Next, let's prepare our folders and files. You may need folders for the raw data, the FASTQ files, results, plots, libraries, and so on. Create them accordingly. There are three inputs we need: the sgRNA library file, the control sgRNA file, and the FASTQ files. This is the format of the sgRNA library file. There are three columns in this file: the sgRNA names, the sgRNA sequences, and the sgRNA targets. These columns are separated by tabs. pay attention to the sequence column. Sometimes you get RNA sequences instead of DNA sequences, where Ts are used. If this is the case, make sure to convert them into DNA sequences using seq-tag, or the pipeline will fail. The control sgRNA file only has one column, which contains the names of the negative control sgRNAs. Now let's count our sgRNA reads. In our MAGeCK environment, call MAGeCK count -L to specify the sgRNA library text file, -N to give a name of your run, sample label to label your samples, specify your FASTQ files. Note, the order of the sample label must match the ones for the FASTQ files over here. The output of MAGeCK count is a count table in this format. Looks familiar? Yes. It's exactly the same format as the count table in RNA-seq data analysis. With the count table and sample grouping information, we're ready for the differential test using MAGeCK test. -K to specify the count table from the last step. -T, the label of the treatment. -C, the label of the control group. -N, give your comparison a name. Control sgRNA, specify the control sgRNA file in the previous steps. The gene LFC method uses alpha median, and norm method uses median for robust test. The output files include a gene summary file. This is our major output. An sgRNA summary file, and some R scripts. You can ignore them safely. This is the format of our gene summary file. The columns are self-explanatory. Genes are ranked by negative selection over here. You can re-rank them by positive selection. The top-ranked genes are always informative. If you decide to go further, we can visualize the results from MAGeCK in R. Open your RStudio, load the essential packages. There are 3 output files from MAGeCK we can explore. The count summary file contains some QC metrics. The gene summary file contains our major results. And the sgRNA summary file contains the sgRNA performance data. We can visualize specific columns of the table using ggplot2. This is the syntax of ggplot2, where lines are connected by plus signs. Each line represents a layer of the plot. For example, the first line represents the data layer, where you specify which dataset and the columns you want to visualize in the plot. The second layer is a plot type layer, where you specify you want a bar plot, box plot, violin plot, or something else. All other layers are pure aesthetic. Let me show you an example. Say we want to plot some QC metrics from the count summary table. First, we read in this table, which has the following columns. We want to plot the label column and the gene index column. So in the first layer, we specify the dataset we want to plot. We want label column in the x-axis and gene index in the y-axis. And then we want a bar plot. Then we add some aesthetics and annotations to the plot. This is the final plot we get. We can do the same thing for other columns. As you can see in our case, sample 1 may have some problems, but they're not serious. With the same logic, we can plot top ranked genes, top positively selected genes, top negatively selected genes. We can directly compare the total number of positively and negatively selected genes. We can also compare the performance of the sgRNAs for top genes. In CRISPR screening, we usually use multiple sgRNAs targeting the same gene. As you can see, their efficiency are not made equal. And this is all I have for today. Subscribe if you like it, and I will see you in the next video.